Circadian rhythm research is key to understanding sleep disorders and neurological diseases affecting millions of people around the globe. But scientists face a massive bottleneck. They must analyze thousands of neuronal cells across days of imaging data, manually tracking their activity frame by frame. What could be taking them minutes is currently taking them hours to days. Dr. Allen's lab at Oregon Health Sciences University spends countless hours on this tedious process. Their current methods of tracking is error-prone, limits experiment scale, and worst of all, delays critical discoveries. Researchers waste time on repetitive data processing instead of the actual science to help people. Meet Neurolight, intelligent automation for neuronal image analysis. We can start by opening up the application, and we'll be greeted with the experiment manager. Here we can start a new experiment, enter the name, analysis type, who the principal investigator is, and go ahead and create it. Next up, we can load in our image stack. This is typically 250 to 300 different TIFF images over the course of a week. We enter the time in which the first image was taken, and we can get going. Next up, we can edit the image, ensuring optimal visibility on the two areas we're looking at. Those are the two spherical areas, which are bundles of neurons that we're trying to see if they have a standard circadian rhythm. Once the image has been edited, we can go ahead and align them. We have a variety of alignment types, but rigid body is the best in this case scenario. The reason we need to align these images is because over time something called microscopy drift happens. This is where the neurons will shift around, leading to inconsistent analyses. Then, once the analysis is done, we see the confidence interval, as well as are able to load the newly aligned images into the viewer. Next, we outline the two halves of the SCN using our ROI selector. This is a built-in pen tool where the user can select and create a spherical shape around the area of interest. They can adjust it as necessary, as well as select the other half, ensuring a proper analysis. Now that we have selected those areas, we need to detect the neurons within those areas. As we can see on the left side, we have a variety of settings that we can change to ensure the best type of analysis. We can then see all the neurons that we found throughout the entire time series. The first graph is a standard ROI intensity where we can see over time how bright the areas are. Next are the trajectories where we can see the fluorescence intensity of each individual neuron, and as we can see, it follows a standard sine wave indicating a standard circadian rhythm. We can zoom in on the graph, drag around, show the peaks and troughs. Next up, we have the Lom's-Gargle periodogram. This is useful for neuroscientists to be able to analyze the frequency and period over the time. Lastly, we have the Rayleigh and Rowe analysis. This is where we can see the peak of each individual neuron along a 24-hour clock. What took hours to days can now take minutes. Analysis that was bottlenecked can now happen almost instantaneously. We're cutting analysis time by over 50%, allowing labs to scale experiments and accelerate their discoveries. Neurolight frees researchers to focus on what matters, understanding the biological clocks that govern human health. Visit the link on screen for easy access to our capstone website so that you can track progress on our project. Let's light the way for faster neuroscience research.